Hi guys, welcome back to the Experimental Design and Analysis section of the Core Skills course. This is lecture 6 in the content that I'm going to be delivering, and we're moving on from last week's lecture, which started to look at the assumptions of parametric statistical tests, to look in more detail at those assumptions and in fact a whole bunch of other assumptions which you maybe weren't even aware existed. Before we do, just a reminder from this first slide, it's my kind of biological spot the difference game. Maybe not so difficult this one as some of the others I've shown you. A reminder that we're dealing with differences here. As an example, testing for differences as an example of the kind of statistical tests that we do. We're going to move to look at trends in the next lecture, in fact. Trends being the other kind of major pattern that we look for in the life sciences, differences and trends. So in this case, what you've got on screen are two animals that are clearly different, but what you may not have spotted is that the animal on the left is actually a spider. It's a spider pretending to be an ant. It's pretty good at it. It looks like quite a compelling ant. On the right hand side is a fly. I think it's fairly obviously a fly to most of us, but it's also pretending to be a spider. So if you look at its wings, they form the shape of the front legs of a spider, and the body then sort of becomes the body of the spider, pointing backwards relative to the head of the fly. So these are other examples of mimicry in the natural world, which is this subject that I find so fascinating and is my own research topic, but doesn't need to bother you, the fact that one of these is a spider and the other is a fly. More important stuff to deal with. So let's look at the outline for this lecture, again in 3 parts. This lecture is all about the assumptions of the t-test as an example of a parametric statistical test. And we've already dealt with the assumption of independence in the previous lecture. We're now going to look at 2 other crucial assumptions: the assumption of homogeneity of variance, i.e. the idea that the variances of the samples in the test are the same. That sounds a bit daunting, but it's actually quite straightforward. And then we'll look at the assumption that data are normally distributed, which I've already mentioned, and in particular what to do if that's not the case. One of the things we can do is we can transform the data so that they are now normal, having not been normal. So we'll look at how we could try that. And then if that doesn't work, or we violated the assumption of homogeneity of variance, for example, we might be compelled to do a non-parametric version of the t-test, and so that's what we're going to look at in the final part of today's lecture. So, first part is all about homogeneity of variance. This is one of the key assumptions of parametric statistical difference tests like the t-test. And just to put this in context, this is the second assumption we've looked at, the first in the previous lecture being the idea that the observations are statistically independent. So homogeneity of variance, it's a horrible sounding concept, but actually it's pretty straightforward. When you're running a difference test like a t-test, you typically assume that the variance, the degree of spread in each of the samples is the same. So if we plotted the samples on a frequency histogram, we're expecting the degree of spread of the bell curve to be similar in each of the samples that we're comparing, and not to see something like this where one sample is very variable and another sample is not very variable. In that situation, we would have violated the assumption of homogeneity of variance. Homogeneity just means sameness, as opposed to heterogeneity, which means kind of differentness. So that's the assumption we're making in a t-test and other kinds of difference tests. And we, first of all, really need to check it. And we could eyeball the data on a graph, but often it's quite hard to see what's going on. And anyway, just eyeballing data, we've learned so far, really we need a more objective measure of what's going on. So we could actually perform a statistical test, which is itself designed to check for homogeneity of variance. So that test is called Levene's test, and what you're going to encounter today and over the next few weeks is quite a few test names, unfortunately. They're often named after crusty old scientists from the 20th century, but it is just a sort of a necessary hazard of the job that you come across lots of different test names. So Levene's test is a test of homogeneity of variance. It's actually quite like the difference test, the t-test that we've already encountered. The t-test is looking for a difference in means. Levene's test is looking for a difference in variances. So it's not really all that different from a t-test itself, but it's testing for a difference in variances. And the null hypothesis for Levene's test is that the sample variances are identical. In our case with the t-test, we've got two samples, so the assumption is that they are the same or not different. And we run Levene's test. We can run it using any of the sort of modern stats packages on a computer, and just like other stats tests, just like the t-test, it generates a test statistic, which for the t-test was called t, but for Levene's test is called f. And that test statistic has known mathematical behaviour when the null hypothesis is correct. So if we know how it should behave when the null hypothesis is correct, we can ask, well, what's the probability of observing the variances we've observed if the null hypothesis is correct? In other words, we can calculate a p-value. And that's the whole point of doing the test in the first place. So we're going to generate a p-value for Levene's test, which is going to tell us whether or not the assumption we were making when we were about to run a t-test was correct. So it kind of all feels a bit meta, this. We're doing a test in order to find out whether we can do a test, but that is the nature of the beast. So let's look at it in the case of a real biological example. And this is a nice behavioural example from snails. Snails have quite peculiar sex lives. You may be aware that the snails are typically hermaphrodite, so they are able to produce both male and female gametes. And so that means in theory that they can self-fertilise. So they could get by without a sexual partner. They can basically have sex with themselves. But all things being equal, they would rather find another snail to exchange gametes with. And you can think about that in terms of the advantages of sexual reproduction. Mixing your genes with another individual gives your offspring more variation and more opportunities to exploit the environment in lots of different ways. There's a whole field of literature about why animals and other organisms tend to be sexually reproductive rather than asexually. But anyway, these snails have the kind of the joker card up their sleeve, not that a snail has a sleeve, that they can opt for self-fertilization if they need to. And that might be because there aren't any sexual partners around, or it might be because they're worried about dying. So if you're a snail, there's a hypothesis embedded in this study that I'm going to tell you about, that if you're a snail and you're scared of dying, then you might be better off getting on with reproduction now. In other words, not searching for a sexual partner, but instead self-fertilizing and laying some eggs in case you get nobbled by, for example, a predator. And the experiment in particular that we're looking at is one where they exposed these aquatic snails, they live in fresh water, to some odour from a potential predator, in this case a crayfish. So crayfish is a crustacean that likes to eat snails. And we're looking at an experiment where one group of randomly selected snails are exposed to the odour of the predator, and one group are a control group. And then we look to see how long it takes until the snails fertilise themselves. And in the control group, I realise I've missed the units off this graph, that's terrible. In the control group, it takes 4.5 somethings. I don't know whether, I think that might be days or hours. Whereas in the group exposed to the predator odour, that seems to be much reduced by more than 50%, in fact. But obviously we need to test that statistically to be sure. But the error bars give us a clue, but they also give us a clue to something else. So the sample size in each case here is the same, but the error bars are very different. The error bar on the left here for the control is quite large, suggesting low confidence in where the mean is. And on the right it's small, suggesting high confidence. Now, if the sample size is the same, then the only reason why we lack confidence on the left but not on the right must be because the data are more variable, and indeed that is the case. The underlying data on the left here in the control treatment were more variable. There was more variation in how long it took for snails to fertilise themselves than on the right, and that suggests that the assumption of homogeneity of variance might be wrong. So before we go ahead and do a t-test, we need to check that assumption of homogeneity of variance. So we run a Levene's test. It generates a test statistic, in this case f, 25.64, and a p-value of less than 1 in 100, less than 0.01. What that means is we need to reject the null hypothesis of the Levene's test, which is that the variances are the same. So we conclude that the variances are significantly different, and that means we've got a problem because the t-test assumes that the variances are identical. So if they're not, and we've got strong evidence they're not, then what do we do? Well, oh sorry, this is just repeating what I just said, that we've rejected the null hypothesis of the Levene's test. The variances are not the same. Oh, and there's a crayfish to show the kind of thing that eats snails. So what we need is an alternative strategy. And in the case of the t-test, there's a very straightforward alternative strategy, which is a slightly modified version of the t-test called the Welch's t-test, which is available in most standard stats packages. Sometimes it isn't even given that name, it's just called a t-test with unequal variances. So for example, you can even run this in Microsoft Excel. You can run a plain vanilla independent samples t-test, or you can run Welch's t-test, only in Excel it's called just t-test with unequal variances. So when you do that, the analysis makes an adjustment for the fact that the variances are different, and you get a reliable result as a consequence. Okay, that's the end of the first section of this talk, which was about the assumption of homogeneity of variance. In the next section, we're going to look at the assumption of normality, which I've talked about already a bit in this course.